So the plan for today is we need to finish off the lecture stuff. So we need to talk regulation of pain and then temperature receptors. I don't think those will take too long, especially the second bit, but I think it's interesting, worth covering. After that, we will get into the homework. When we return, please remember there's a quiz on Chapter 7 that is due Monday night, 11:59 p.m. So that's not this coming Monday, that's a week from this coming Monday. So you can wait if you wish until you get back on campus after spring break. Or you can do it anytime in between now and then. And then we will be starting into the Chapter 11 material on the vestibular system. So you should look for slides and such to appear sometime over break, assuming I get them done then. Any questions? Oh, and then also, of course, the paper summaries are due today. That'd be the one other thing I didn't put on there, but that's important. Anything that you're wondering that I didn't talk about? You look. No, just like, it could be like a panic thing, or it could just be a, like, all right. So, well, let's get back to this. So we were talking about how pain information is transmitted up to the brain, and we went through the spinal thalamic pathway. What is left to talk about pain is to talk about to talk about how we regulate pain, because pain is not really a constant impulse that's always consistently transmitted up to the brain. We actually modulate pretty far down in the spinal cord oftentimes pain signals coming up toward the brain. And so as a result of this modulation, we may be able to perceive more or less pain depending on whether or not these systems are engaged. So there are three main ways that pain is regulated. The first is discussed on this slide. It's called afferent regulation. So afferent regulation basically refers to the idea that there are multiple afferent signals. So afferent signals are the signals coming from the peripheral nervous system into our central nervous system, like an afferent neuron. And these afferent signals can effectively kind of cancel each other out. So specifically, the big picture idea with afferent regulation is that your pain input can be modulated by your touch input. So if you have pain input just coming in by itself, pain input will be generally relayed directly up to the central nervous system via the spinal thalamic pathway that we just talked about last time. However, if there is simultaneous pain and touch input coming in from the same general location, the touch input can literally kind of deaden the pain. So I think this is a big part of what's going on when, like if you get injured or hurt, sometimes people talk about you sort of rub it off or you walk it off, and that can actually reduce the amount of pain that's going. And one of the things that's going on is that your touch input can deaden or minimize the amount of pain transmission that's going up into your brain. Here is a way that it works. This is often called the gate theory of pain. So here what they're depicting, this is the edge of the spinal cord. So this big picture is a blow-in version of this little box up here. So here's the spinal cord. This would be dorsal horn of gray matter. So this is the spinal thalamic pathway, basically. Here's your C fiber carrying pain information coming into the spinal cord. And what we talked about last time is that this C fiber would then form a synapse here within the dorsal horn of the gray matter, and then give rise to a second order neuron, which would decussate and cross over and head up toward the brain. And so if all that is happening is the C fiber is bringing in its signal, you'll see there's a little plus sign here. That's indicating that the end of your first order neuron is stimulating your second order neuron, and the signal goes on up to your brain. So that's just right what we saw last time. The new thing that's in this figure is that they're also showing an A alpha or A beta axon. So this would be a touch receptor this would be an axon coming from a touch receptor somewhere that would be basically coming from the same location where this pain information was coming from. And so if you follow along with the circuit, what it's showing is that this touch receptive axon, in addition to sending a signal up the dorsal column, because this is going to go up the dorsal column, medial lemniscal pathway up to the brain, it's also forming a synapse here in the dorsal horn of the gray matter. And the synapse is on an interneuron. So if you have touch reception in, you will stimulate this interneuron. That's what the little plus sign says. And then what would happen is this interneuron would inhibit the second order neuron in our pain pathway. So again, if the pain input comes in by itself, you have immediate stimulation of your second order neuron, pain information goes up to the brain. But if at the same time you have touch information coming in through this blue neuron, the A beta neuron, that'll stimulate an interneuron, and the interneuron will then inhibit the second order neuron, and so now your second order neuron in the pain pathway is kind of getting turned off, or at least it's being inhibited, so that it's not going to be as responsive to the stimulatory input coming in from the C fiber. This might not completely eliminate the firing of the second order neuron, but it might reduce how much it's firing. So you're reducing how much pain you're feeling by having touch input coming in at the same time as the pain input. So I'll read through the slides, which basically says the thing. So a second order spinothalamic neuron is affected by two neurons. One would be, so this second order neuron, that's this one. One would be the nociceptor neuron, that's your C fiber. The other would be this inhibitory interneuron. So the C fiber, if excited alone, you have the pain receptor the pain receptor actually inhibits this interneuron a bit, but it's going to stimulate the second order neuron. But if the A beta axon and the C fiber are stimulated simultaneously, that should result in the A beta axon exciting the interneuron, which will then inhibit the second order pain neuron and prevent it from responding vigorously to whatever the C fiber is telling it. So there's a way in which this kind of functions as a gate. So we can kind of close the gate by having touch reception come in at the same time as pain reception, and therefore prevent our second order pain neuron from getting excited. Does that make sense, more or less? So this is a reason why it really does sometimes help. If you get, like, you know, I don't know, I have baseball in my mind. So if you get hit with a baseball, sometimes literally just kind of rubbing it really does help. It reduces how much pain you're feeling, and it's that inhibition of pain. It's not just in your head. It's actually not in your head at all. It's actually happening in your spinal cord. Isn't that cool? Yeah. Yeah. Well, so it could be like if it's immediate. So any sort of immediate help with the pain could be a result of the massage. The other thing the massaging sometimes is doing is it's helping to kind of move fluids around. It's kind of helping to spread around inflammatory compounds, which can ultimately, in the end, reduce inflammation. So that's like long term, I think that could be what massaging an injury is doing. But like if it's just like this immediate thing after you got hit in the arm and you just kind of do this and it feels better, that's this gate theory. All right, questions? Is that all right? Cool. So that's one way we can regulate pain. Here's another way. This is called descending regulation. So if the last one was about two afferent inputs kind of talking to each other and one's negating the other, this one is about descending regulation. So that is information from the brain traveling down, descending down from the brain into the spinal cord and basically shutting down pain pathways. So there are many ways this can happen. We're going to kind of talk about, I guess, sort of just one. But the idea is that your state of mind, might be a way to think about this, can dramatically affect your perceived pain. So if you are in a state of excitation, you've got your sympathetic nervous system running, your fight or flight system is going, like maybe you're exercising. Maybe you've had this sort of thing where you'll be exercising or, I don't know, just like having fun running through the woods or something, and you get scraped, and you don't even realize that you got scraped. You don't even feel the thing. And then you get done, and you look down, and you're like, oh, I got scraped. And then all of a sudden, you know, maybe you can start to feel the pain too at that point. But you didn't feel it at the moment it happened. In part, what might have happened to inhibit that pain signal when you got scraped is that you had descending regulation going on, where your state of mind, you were excited or stressed or something, was causing you to repress pain. The way it seems to work is that information from your emotional centers within your brain, and other kinds of inputs as well, may feed into areas of the brain like the periaqueductal gray matter. So this is a part of your midbrain that we saw when we talked about midbrain slices. So other things we talked about, well, there's the substantia nigra. This one doesn't show the red nucleus because it's a slightly different slice. But this periaqueductal gray is a sort of band of gray matter that's right around the cerebral aqueduct, which is right there, that little hole. And this is an area that can get input from other brain centers, higher brain centers, Could be cortex, could be hypothalamus, could be other parts of limbic system. And when excited, it'll get neurons excited that send excitation down axons that travel down through the medulla, often going through the Rafe nuclei, which are found right here, sort of, right in this deep part of the medulla. This is just another collection of gray matter. And then here's another neuron that will travel down into the spinal cord. And so what will happen is this axon coming out of the Rafe nucleus comes down the spinal cord and may inhibit some of these second-order neurons that are part of the spinal thalamic pathway. So the inhibition is not coming from interneurons that are being stimulated by outside, like, input, like touch input that's coming from outside the nervous system. What's happening here is your brain is basically repressing your pain by sending signals from the periaqueductal gray through the Rafe nucleus down into your spinal cord to basically shut down your pain pathways. Here's another picture that kind of shows something like that. So this is from a different book, but excuse me, I thought it did a nice job of kind of illustrating it. It doesn't point out the same exact structures. It just sort of refers to them as midbrain and medulla, but this would be where your periaqueductal gray would be, and then the Rafe nucleus is down here in the medulla oblongata. So you can connect these to the prior slide, I think, pretty nicely. It's just a good demonstration. So here, this would be your hypothalamus, this would be your cerebral cortex. There could be other brain centers, limbic system centers, for example, which are your emotional centers, sending information to the periaqueductal gray, which then sends signals down to the medulla, to the Rafe nucleus, which then sends signals, this is going down this reticulospinal tract in this case, that are causing inhibition of the neurons. So here's this axon coming down, and it's now inhibiting. Here's a nociceptor C fiber coming in, and it's being inhibited by this signal coming from the Rafe nucleus. So that's that big negative sign here. Okay, so it's basically showing the exact same thing, just a sort of different rendition of it. I guess here they're also showing a different, this would be an interneuron that's also inhibiting, this would be the second order neuron coming in with carrying pain information. Again, same basic depiction of what was on the prior slide, but I don't know, somehow I like this one a little bit better, although it doesn't identify the anatomical structures as well. All right, that's number 2. Number 3, third way to regulate pain, endogenous opioids. So this came up on the homework, at least in part. But the idea here is that we also produce opioids, which are compounds that stimulate opioid receptors. Now, opioid receptors are famous because they were discovered through, basically through narcotics, because these are receptors that exist within our brain that when you take something like morphine or codeine or heroin or any of these other various opioids that exist either on, either through prescription drugs or through black market, they bind to these opioid receptors. The job of these opioid receptors, they're not there, though, just so we can respond to morphine. Morphine is a compound derived from a plant, but then it happens to bind to these receptors, and that then leads to the strong reaction that we have in terms of pain inhibition that we get from taking morphine. The way it works is that normally, in our normal brain, when we're not taking morphine, we produce what are called endogenous opioids, which are compounds that are kind of like morphine or codeine in their molecular structure. And these are the things that these opioid receptors that are produced normally in our brain are supposed to bind to. We produce them, again, kind of depending on our particular state of mind. So we tend to produce more endorphins, more endogenous opioids. These are the same things as endorphins. When we're excited or when we're nervous about something. It's not the same mechanism here, though. We're not sending neurons down periaqueductal gray to the Rafe nucleus. We're producing these basically as hormones or local neurotransmitters. And they're delivered into some of the pain regulation areas. And it says, what areas would you expect? I guess I'm not going to ask you, but these are these same kinds of pain regulation areas we were just talking about over here. So they're interfacing with some of the same signals. But again, this is different. This isn't worrying about axons traveling from these brain regions to another region. It's not a neural signal. It's kind of, you can think of it as almost like a hormone, or at least a local hormone that's being released and bathing the medulla or bathing the areas of the midbrain that are related to pain with these opioids. And what they do is, when your endogenous opioid receptors are binding your endogenous opioids, we tend to reduce the amount of pain transmission that is permitted to go up through your spinal cord, through your midbrain, and ultimately up into your cerebral cortex. So the way these things work is they do suppress glutamate release by your nociceptor neurons. So if there's less glutamate, less neurotransmitter being released, you'll have less signal being relayed up to your cerebral cortex ultimately. They also can directly hyperpolarize nociceptor neurons. So they're making them more negative, which means it's harder for them to get up to threshold, and therefore requires a larger than normal stimulus to get them up to threshold. So this is what they're supposed to do. Now again, of course, with illegal narcotics, so if you're taking heroin, what happens is you get this huge bolus of compounds that are binding to your endogenous opioid receptors all at once, and certainly that's going to remove lots of pain, and also, brings with it feelings of just, I guess, incredible release and such. I haven't tried it. Questions? We'll come back to, I think this is really interesting because that question about placebo, and so we'll come back to it in the homework. All right, last little thing that I want to talk about then is temperature perception. We're not going to really talk about the signaling system, although this is basically carried also along that spinothalamic pathway, so it kind of does the same thing pain reception does. But I do want to talk about how temperature receptors work, at least in passing. So we have some sort of brief acclimation to them. Acclimation, it's kind of funny. Okay, so the idea with temperature receptors is that all throughout our skin we have numerous neurons that are sensitive to changes in temperature. And the way they work is that their membranes have particular proteins that will change conformation depending on the heat environment around them. So the more heat there is, the more they tend to vibrate around, and the more they vibrate around, the more they will tend to change from one shape to another. And this can cause them to open or close depending on which particular one we have. We tend to have lots of them. So we don't have a situation where there's like one temperature receptor that opens more and more and more and more as we get hotter and hotter and hotter. Instead, the idea is that as you go from sort of having cold exposures exposed on your skin to very high temperatures exposed on your skin, you will transition from basically having different sets of these temperature receptors being excited. So what this is trying to show you, so this might be the membrane of a neuron that is temperature sensitive. And it may have several different receptors that are embedded in it. But the idea is that each of these receptors is going to be reactive in only a small range of temperatures. So for example, here we are at, this is 0 degrees Celsius, so this would be 32 degrees outside. So this is roughly what it is right outside right now. Going on up into like 40 degrees Celsius, this is like, I don't know, was that like mid-90s or something Fahrenheit? And then this would be getting up even higher. So 50 degrees Celsius, that's halfway to boiling. So that's getting well into the hundreds. What you end up seeing is that if you're going outside right now, the main receptors that would tend to be excited would be this one called ANKTM1, and then also this one TRPM8. Basically, the higher the line is over here, the more channel activity, and therefore the more this is going to stimulate the neurons that happen to have these particular receptors embedded in them. So as you so if you start here at 0 degrees, at freezing, these guys tend to be very, very excited. But as you shift up in temperature, so as you go toward sort of common room temperature, somewhere around like 20 degrees, once you get to there, you still might have a little bit of TRPM8 activity. So that would be this green one. But ANKTM1, those channels and the neurons that have those channels would basically be shut down. They're not going to be firing anymore. So you can perceive the difference between cold and room temperature in large part because you no longer have these guys firing at all. And so your brain says, all right, so it's not cold anymore. These guys might still be firing a bit, so it's not super, it's not, it's still kind of chilly, or at least room temperature-y. But as you shift up, So as you get up into like the high 20s, you get into a point where none of these channels get excited. So you kind of have no input. This is basically really, you know, I tend to find mid 20s is a pretty comfortable temperature, and it's something like around 80-ish or something. If you know your Celsius better than I do, you can just sort of smile and, you know, nod in derision, or, you know, shake your head in derision, because I'm an American and I don't know how to do Celsius very well. Once you hit 30 degrees Celsius, though, you'll then start to have other receptors that start to open. So other channels, so like this one, TRPV4, starts to open around 30 degrees Celsius, and will open more and more and more until you get up towards 40. So this one will increase its activity. Once you hit about 35 degrees, another one engages, TRPV3. If you get really hot, so here we're getting towards, you know, this is like 100 degrees or so, TRPV1 starts to open. And then if you're crazy, like dangerous hot, that's when this TRPV2 one starts to open. And so you'll have, the big picture here is, I'm not going to require you to I'm not going to give you a multiple choice question without this picture and say, at 35 degrees, which receptor is activated? Like, that's not how I want you to do this. I guess I could give you this and then ask you to do that. I want you to be able to like look at the graph. But the point is that at any given ambient temperature, a different suite of temperature receptor channels will be open. And so we can kind of figure out what the temperature is based on which ones of these happen to be open and active at a given moment. Does that make sense? Yeah. Oh, sure. Yeah. So, you know, you'll have some neurons that have TRPV4 channels. You'll have other neurons that have TRPV3 channels. And so at, you know, this sort of, let's go with like right here about 38 degrees, both of those would show activity. But the neurons that have TRPM8 and ANKTM1 would not be active. So we can tell what the temperature is from that. I think it's based on the experiment these were recorded in, so the dotted lines are more of like an estimate or extrapolation for what we think they are. I'm not sure if that really is uncertain or if it's just a matter of they were producing this from some source where it was not demonstrated in that particular study. So this is the idea, is that you've got different temperature neurons. The different temperature neurons are distributed around your skin. Different ones will have different channels, so they're reactive to different temperature ranges. And so the suite of signals that are sent up to your brain, based on that information, your brain can decode that based on which neurons are firing, and you can kind of triangulate what the temperature is like. If you've ever studied color perception, so the way we tell blues from reds from greens when we look at them through our eyes, it's a very similar kind of a thing where we have some neurons, effectively, some cones that are sensitive to blue light, some that are sensitive to red, some that are sensitive to green, and we figure out what the actual color is that we're looking at based on how much the blue receptors are excited versus how much the red ones are excited versus how much the green ones are excited. So it's the same kind of principle for what's going on here. That won't help you if you've never looked at color vision before, but that's okay. These signals, whatever the neurons are, and whichever channels happen to be excited, they will be sent up either A delta or C fibers up the spinal thalamic pathway, so it works basically in the same sort of system. The last thing I want to point out is that some of these really hot channels, I almost said receptor, but these hot channels, the TRPV1 and TRPV2, these are temperature receptors. They also are found on nociceptor neurons. And so, and that kind of makes sense, right? Because especially TRPV2, this one is only going to be open when you're dealing with what are becoming dangerously hot temperatures that could burn you. And so you perceive it as very hot through the temperature sensitive aspect of it, but it also can be interpreted as danger, pain, you know, noxious stimulus through the nociceptors that have the same kinds of channels on them. And of course, there's a little chili pepper because it interacts with these guys too, if you like spicy food. That's a homework question. I'll get to that in a sec. Yeah. Yeah. No, I mean the environment. So this is how you perceive what the temperature is that you're touching or feeling. So you go outside, it feels cold. You put your hand on a warm surface, it feels warm. It's through these. It's a good point, though. Like, I would presume we do have internal thermostat sensors that are detecting your blood temperature, and I would presume they're using the same kinds of channels. I don't know that for sure, but I would expect that's true. Last little ditty is just to point out that these temperature receptors that we were just talking about, these tend to be extremely phasic. So they tend to adapt or acclimate to signals pretty quickly. So what this is showing is an experiment where we're starting with where the skin is touching a surface that is 38 degrees. That's fairly warm. That's basically body temperature. And then it drops down to a bit below body temperature, 32 degrees. So we basically, expose you to a cool piece of something. And then we warm you up again or expose you to a warm thing again. And so what you can see is that initially when you're warm, the cold receptor, which is what this recording, these are action potentials from a cold receptor. They're not really firing. As soon as you shift down to the colder range, that causes those channels to open and you see lots of action potentials fire. But then those channels kind of acclimate and they start to close and they become, this temperature receptor behaves in a sort of phasic manner, so it kind of slows down its firing. It never really stops, but it slows down. And then when you bump back up to 38 degrees, of course, it goes silent. Similarly, here in this line, we're recording from a temperature receptor, or a warm receptor rather. Initially in this experiment, it's going to be firing pretty steadily. You drop down to cold and it kind of shuts down. Once you kick back up to the warmer temperature, it fires like crazy for a little bit, but then again, it's fairly phasic, and so it tends to kind of bottom out. This is basically what's happening as you're I equate this to like getting into a swimming pool or getting into a hot tub. So as you're getting into a swimming pool, you touch the cold water, and immediately it's like, ah, that's cold. And then, a little bit of time goes by, and your cold receptors stop firing as much, and you're okay. Like, it's fairly comfortable. Similarly, you get into a hot tub, whoop, you bump up to a higher skin temperature, and you're like, ah, it's hot, but then again, you adjust. So that change in temperature that we perceive is in large part a function to the activity of our temperature receptors going inactive or acclimating or adapting to the signals. Any questions? Excuse me. All right, so what I'd like to do then is, that's it for a lecture bit. So let's pull out our homework and start working through these. Give you guys, I don't know, let's see if we can start talking about them in about 10 minutes or so. I'll assign questions and we'll start working through them. Not every, like some of these I think have discrete answers, some of these don't, for this one in particular. And that was sort of on purpose. I don't want to accept, I want to track. Okay, so let's start off. So Billy, you got the first one. So describe the woman's primary symptoms. Like, what did you? What was the last thing you said? Coordination and proprioception. Yeah. And her. Oh, right, and general posture. Yep. Yeah, it's almost like, so you probably had the thing, maybe it's just me, but you wake up and you can't feel your arm. And that lasts for a few seconds usually. Probably because you've been compressing your brachial plexus or something and just been compressing those neurons. But usually once you start moving, it's fine. But the feeling just never comes back. So like she's just doesn't feel that anywhere across her whole body permanently. So that sucks. So why did authors of the story test the operation of her parietal lobe? Olivia, what'd you? Okay. So why would you look in the parietal lobe for that? Like, what's the... if you're interested in not getting touch, like what, I guess I'm just asking, what's the connection between the parietal lobe and this kind of sensation? Yeah, so this is like, so the parietal lobe's where the primary somatosensory cortex is and all. I was probably asking too simple a question, but yeah, that's all it is. It's just the parietal lobe's where you process all this info. So you look in the parietal lobe because that's where this kind of, I'm even going to say somatic sensory information should be delivered, is to the parietal lobe. Let's see, 1C went to Mary. So why might she still have light touch perception but not proprioception? Yeah, maybe. I'll put the next one down. So what do you think is the apparent cause of symptoms? There's no right answer to this one, but what'd you put, James? Trigeminal touch. What was it? Spinocerebellar? Can you justify those? Like, why do you think one versus the other? It is. Especially cerebellum, I think. So like the thing about the spinocerebellar tract is that it's talking directly to cerebellum, and of course coordination's where cerebellum is. Mary had a different, I'm just going to put it up there because you mentioned it. Another explanation is area 3B or 3A might be damaged. So if area 3A is where all the proprioception is arriving, that could be a problem. You know, so if there's damage there anyway, that would mean that you might not have proprioceptive ability anymore. Whereas, you know, the touch information that goes to 3B, at least some of it seems to be getting there. I think she lost kind of like the pressure reception. So that's kind of fouled up too. But so like the Pacinian corpuscles, but there's this bit about she liked to go in cars and feel wind on her face because she could actually, or on her body, because she could actually feel that still. So that bit that's going to area 3B is still getting through somehow. Anybody have any other causes you picked up on? Yeah, would you? Do you mean like in brainstem or like more nerves coming into the neck? Okay. Okay, so these would be like spinal nerves outside of the central nervous system. And if, you know, up around the neck, especially as you get down from the neck down lower, you start to run into this brachial plexus, which is where all the neurons that are servicing your arms are. So if that was inflamed, at least that would explain the loss of hand control. Maybe not the legs, but the hands it would explain. Did I see peripheral? Yeah. I missed that. Okay. Yeah, I don't remember that, but I'm not doubting you. I don't remember that. There was definitely discussion of like a neuritis, which would be inflammation of nervous system tissue. This is terrible. I didn't read it lately, so I'm trying to remember where they said that. That was either brainstem or that was spinal cord, and I can't remember. Does anybody remember? Okay. So, so the sensory root would be the dorsal root of the spinal nerve, or the posterior root. So this is where all the sensory information is coming in. So it could be before you even get into the brain, and some of our brain areas aren't directly involved, but it's sort of preferentially affecting those neurons that are bringing proprioception in. There's no resolution to this. Like, we don't really get an answer. The reason I threw it in here is because I did like that it did sort of identify that there is different processing of your light touch information versus your proprioceptive information. So I was kind of keying in on, dorsal column pathway versus, and well, not so much dorsal column, but like this area 3A versus area 3B difference in terms of where these areas are processed, because that does at least explain why she could still have light touch, but she wouldn't have proprioception. But we don't have a final answer. I was just, I think it's kind of a fun case. But have anything else they want to throw in on this? I think I got all the hands I saw, but okay. Sorry about the dinging. That's my boss. Okay. Summarize so number 2. This is where all we did was talk about the spinal pathways carrying information in from the body, but we didn't do anything on information from the face. So that's where this question is meant to come in and fill in the gaps. So it says, summarize the trigeminal touch pathway, identify what the pathway services, location of the neurons in the pathway, where those neurons go, that kind of thing. So this one went to Leandra. What'd you get? Okay. Yep. Well, so if this is touch, I mostly agree. So the touch pathway for spinal, if it's coming from the rest of her body, it goes up dorsal column and then hits the medulla, and so the decussation is actually in the medulla, but it's not the pons, sort of the point. So rather than in the medulla. Oops, sorry, I didn't mean to scroll down. Cool. Can't spell. Let's try to get B in. I think we can do that. Summarize the trigeminal pain pathway. So this is pain instead of touch. What's going on with this one? This went to who? Anna. Sorry, I missed the last bit of what you said. So spinal trigeminal nucleus, the brain stem, then go up to the thalamus. I'll just throw in and then ultimately up to the cortex. As usual, it's the second order neurons that are decussating, as we've seen before. Can we do C real quick? I know we're out of time, but so Taylor, what'd you get for C as far as? Yeah, so I've heard this described as like as suicide disease or suicide disorder. It's supposed to be one of the most painful things a person can experience, where you just, you feel like your face is being electrocuted, and the stimulus for this is like you smile or something, and so you get just the lightest little bit of touch on your face, or you're in the shower and some water hits your face, and it results in just horrible, horrible agonizing pain, just sort of like being electrocuted. But again, it's directly related to these trigeminal pain pathways, and the touch pathways kind of cross-talking and getting messed up because the trigeminal nerve is getting pinched somewhere along the way. All right, I'm sorry I went over, but I wanted to finish off that question. Hope you all have a wonderful break. We will finish these off when we get back on Monday and then launch into vestibular system.